Yeah, thank you very much. Thank you for the invitation for the ACD conference. My name is Thorsten Gressling. Let me introduce short myself. So I'm working for Bayer in Wuppertal, which is a quite remarkable location. So as you can see here, beside the famous Schwebebahn, there are the laboratories on this side, and here we have production. It's one of the seldom constellations where you have R&D and production side by side, and that's very interesting, especially for me as a chemist by training. I love that very much, that you are so close with your colleagues and you can do real intense scale-ups. My other profession, I'm also lecturer at Humboldt University in Berlin for chem informatics and lab informatics. So a lot of activities ongoing. My role is digital chemistry transformation lead. Okay, so let's go to the talk today. So accepting what works, ISA-88 for process chemistry. When I was suggesting this title, there was a bit silence in the room. So, is that really a good title? Yeah, flashy, it's missing. I think that is a real good title because one of my fundamental assumptions is that a lot of good things have been done in the past, but there was not the time to get a good maturity level. And that's why I'm telling this. So it's not always ChatGPT to have the latest, biggest achievements for the audience. There are also things that have evolved over a long time that are working very well. Yeah. So not always ChatGPT, but ISA-88 is also working. Well, what is analytics and process chemistry? Let's ask ChatGPT about that. Yeah. So why do we need analytical data for process development? It's a quite profound answer we have here. So 9 points, and so I can refine. Say, okay, please limit to the 4 most important topics. Yeah, and as you can see, it's also tolerant about typos. I love that very much. And you can see, okay, so for example, analytics is important for process assessment. Yeah, sure, for optimization, identify bottlenecks, inefficiencies in a process. But the most important thing is the bullet point 3, which is decision-making support. That is what we are going, because we go into the direction of data-driven science. And that is because the automation of decisions is getting more and more important. Yeah, we are moving into a direction where not only humans, but also algorithms, and as you can see here, maybe also neural networks become more and more the driver. And that is also for reactions. Yeah. And by the way, why is analytics important for process chemistry? Well, what is the first thing? The molecule or the reaction? Yeah, sure, the reaction. Yeah, and that is why we have to put our focus on that. Okay, let's continue. How is ACDLabs software used in this context? Well, it's a very profound answer. I'm very happy that you are, let me say, rooted in this GPT. ACDLabs software efficiently supports improvement of process based on data-driven insights. That's the last sentence here. Yeah, data-driven insights. And that's what we're talking today. And the final question I gave to ChatGPT is, I need a real-world example with real-world process data. And that's unfortunately, due to confidentiality and so on, I cannot give an example. And that is good. If there would be an example, I would be really concerned. Good. So now let's go into details. What we're talking about, the ISA-88, what is it? Well, if you want to have insights, and especially later on features for artificial intelligence, you need to combine the information you have for the batch, the experiment. That's what you've done on the benchtop or in the kilo lab. And you've combined that with analytic information. And that combined with analytic information is definitely the home turf of ACD, of Luminata, for example. And that's what we're talking about. So the analytic part that is in the middle, well, we'll talk about that later, but we start with the batch data and unit operations, the building blocks. And what has Bayer done here to collect all these things? Are we the first to collect all the batch data across different functions? We got the idea 2 years ago, for sure, to use a data format that, as I said in the beginning, is quite mature. So it's the ISA-88 format, which is standardized since 1988. Yeah, and if you look to a very common blueprint of a unit operation sequence, you see on the left side that, for example, you have things like you put things on a tank, yeah, so raw materials A, B, or C. Then you do the preparation, and after that, from the tank, you go into the reactor, yeah. Also decide if it's reactor 1 or 2, you have the reaction, the chemical reaction, and it's the end of it. And how do you put that into data? Of course, you use this standard data format where you have, it's a classic thing, batch information, which is the process action, the process operation, the process stage, and the process itself. That's it. It's nothing more. Very simple. And you do not need to reinvent the wheel. Okay, so that's why we committed to that and said, okay, is ISA-88, because it's 30 years old, in industry 4.0, yeah? And it says, okay, in 4.0, it aids the integration of data from various sources. That's excellent. This harmonization is crucial for creating a connected and transparent system, one of the key principles of Industry 4.0. Take the first bullet point and you're happy. Yeah, is ISA-88 used in Industry 4.0? Also a typo here. Perfect. Okay, let's commit to that. We have done that. And so I'll give you a short example. Don't be afraid, you will understand that very, very soon. So this is the standard classic file format. Yeah. So this taxonomy that is hierarchical. Yeah. You start in this case with an experiment. You go to the process stages, the phases. Yeah. And for example, for the operation sequence here, you have something like dose. Yeah. Or heat. These are the things that are collected in this data file. Must not be a data file, it's just a data structure. And then it closes, it is a hierarchical thing. This taxonomy closes then at the bottom. And there you have, for example, the chemicals or other meta information you need for the information you have done in the experiment or the batch. That's it. It's quite simple. Yeah, it's a hierarchical taxonomy, and that's why it is such a pretty and wonderful thing to use this. Good. So what is with the ISA-88 architecture we have? Because we just have not, Bayer is very big. We just have not one reactor or one bench top. We have hundreds of them. And so we need an architecture to integrate all these tiny experiments and things. So now a second slide. Don't be afraid. This is an architecture that consists of 4 layers. 4 layers. On the bottom, we have all the things that are carried out on the benchtop, where we have, for example, Siemens or Metal Toledo. We have Systag reactors and things like Lab Operator. Then on the layer that is in red, we have the famous BaseX database, yeah, where you have collected all these ISA-88 information. It's not a file system. It's really a database where you can query and you can exchange nodes. This is combined, because we are in chemistry, with a database. It's called Open Reaction Database. Yeah? If you have information about what is happening in the reactor, you do not have information about the reaction. Yeah? It's chemistry. So chemistry and the what is happening in the reactor is complementary. And then after the red layer, you have the authoring layer. Authoring means here are the receipts created. Yeah, this is where you write your unit operations and what you intend to do and what is later on carried out in the lab. So it's what we call the master recipe. Yeah, and when you get the result with all the time series data, for example, the sensors, I will show you later all the curves, then you have the control recipe. Yeah, and finally, and that is on the upper right here, you have the data science layer. Yeah, and that's where ACD is coming into the game, because here you map. Here you map the analytical information about the process to the receipt, and here you also get the information what has really happened, because here you have the eyes that are looking into the reaction. And then you go with that to data science and later to AI. I will give you also an example for that. Good. Every green bullet is implemented, or let me say at least has an MVP or has a proof of concept. Yeah, so we're quite major here. So let's go to the building blocks. The different vendors, how do they support this ISA-88? I will start for good reasons with the experiment, which in our case is based on Dotmatics. Dotmatics is a ELN that is very, very pretty designed for chemical reactions. So on the right side, you see, for example, the decision tree that leads to a structure, yeah, with the different experiments. But also in the middle, you see analytics, for example, for proven acceptable ranges. We are going into solubility and all these things you need for process chemistry. And by the way, on the lower right side, you also have statistical analysis. Yeah, good. That is the key aspect of the Dotmatics. But you also, when you're creating an experiment, I will give you an example right now. You have, I've obfuscated the structure here. You have this typical sheet of experiment planning. This is the upper part of that, and there you have the lower part of that. And you can see, I will enlarge that here. You have all the operations that are planned to carry it out. Yeah, so you add chemicals, which is the start point, and then you dose later on when you heat and steer. All these things are unit operations we are carrying out. And now what we are doing is we export this. This is an export button. We call it export XML. It's ISA-88. We export that into our infrastructure so that we have the planned recipe. Yeah, so this is, we've seen that before. This is exactly what we now then have into the database. And that's what is carried out by the lab equipment. Next example, what is the building block? Our synthesis automat. One of them is Systag. Systag is a Swiss company creating these machines. These are some photos of that. So pretty sure you're aware of these synthesis machines, of these robots. We have quite major digitalization level here. So for example, we can go on all machines with software interface. This is an example. For example, you can put this also on devices like the HoloLens when you're in the lab. You can control the reactors via virtual screens. That's really excellent, and we can carry out the recipe what we have put from Dotmatics into the Systag and vice versa, and we get out for sure the executed things. Yeah. So this is a very simple example. It's one of the first implementations here. You can see, for example, curves that are out of these reactors. Good, Systag. What we have else? Lab operator. So not everything on benchtop is based on a completely assembled reactor. We have ecosystem, it's called Lab Forward, which is more for the spreaded things that are on the table. So if you have, for example, a stirrer that is not mapping to other ecosystems, or a heater or something like that. This is done by little boxes based on Raspberry Pi. It's very robust, this system. As you can see here in the photo, there's a scale, there's a shaker, and that's also working. And also lab operator now can create out from the many different things we have right here. They can create this ISA-88. So these are the trend data for to temperature, for example. Check. Works. Next one. Okay, Mettler Toledo. They are the pioneers here. Actually, this ACE-88 format were adopted by them many years ago, and I'm very happy, and it's really a great pleasure that they are working in so smart way to share also their knowledge. Yeah, it's really great. I appreciate that. And Mettler Toledo, I will also give a photo here, has also very excellent automated reactors. And also they speak this language to have the control batch, the control data you can get out of that. And as you can see here, it's also based on ACE-88. Also here, you have all the time series. Good, that's it. Just a few examples how the building blocks can work together. We also have a tiny viewer app, so if you're carried out your experiment or your batch, you can look into that. It's very simple. Yeah, you open the experiment in the database and can immediately look into the what we call trends. It's very helpful, and as it is all database based, you do not need to have file handles or something like that. Yeah. Okay, back to our infrastructure. What is missing? Well, as you can see, we integrate a lot of things. Where is ACD coming? Do we agree on all the things we have defined? That's why we and CSL Behring took the Pistoia Alliance into the boat. Maybe you're aware of that. It is unification of pharma companies, non-profit organization incorporated in 2009. And we said, okay, to be sure that all these different things on our infrastructure work together, let's start a work group. Yeah, so it should be not licensed, no one owns the standard. And that's currently working on, and we're very happy about that. So now we have the data, now we have the batch. What is next? For sure, let's take all the analytic data we have and merge that together to get insights. So you start with a simple Luminata record without any eye control or ACE-88 attachments. This is the starting point. The display is very simple. And here you have a button. Maybe some of you has realized what it is. It is called eye control. Here, there it is, yeah, eye control. And with that, you open the door to the new universe I've shown you. So that is the next thing. With the new designed ACE-88 import tool, this is connecting to the database. Here are two experiments you've seen before. You can import the data. The XML then from the database is retrieved. You see also a little bit of the database administration UI here. So, and then there it is. All the trends are imported and displayed. So on the bottom of this screen, you see the trends carried out in an experiment. That's great, isn't it? So now we have all the information. As you can see here, temperature and stirring, pH meter, and all these things. So where is the analytics? For sure, now the next step is, if you do not have already done, you have to insert. We have all metadata, for sure. We have all the metadata, for example, of what you've seen in the receipt authoring. Yeah, that's quite sure. But you also have to map now the analytical things, and for that we need time points. Other call that event frames. That's quite fair. You can call that time points, and you can add them manually. Whenever you have taken a spectrum, for example, what I call IPC and in-process control. But also maybe the software is able to log. Mettler Toledo, for example, can do that when you have some of these events. And you need that later on to put the spectra on it. So you can overlay then on the different points in your batch, the reaction. And for sure, then you get some changes in the chromatograms. And that's going into the direction of the result. So this is the key point, what you see here. You have different conditions, which are the time points you see in the upper screen. So condition 1, condition 2, and so on as time is passing. And on the lower screen, you have the control chart. I will enlarge that. And here in the control chart, you have the integration of the diode array. And as you can see here, concentrations of reactants are changing. Yeah, so in this example, it is increasing, where, for example, one maybe of the educts is decreasing. Yeah, and that's exactly what we would like to have. So we can have our time series combined with the analytical data. And that is the key point for that, what we use in process chemistry. Finally, you have a kinetic plot. So as you can see here, this is an example from ACD, where you decrease the educt, yeah, and you increase the product, for example. And our plan, for sure, is all that is now orchestrated in a very visual manner. You can should then later on export or give the data to Reaction Lab from MATLAB Toledo, where you can do real kinetic calculations and then prove if your assumptions for the schema are correct or not. That is really great. Okay, so what is the difference to PAT? Many of you know that there are also major applications of very dense time, let me say, enlarge this here. When you have a time axis and you observe the spectra, you can have that in a very, very dense way. So that is the difference to what we have done here. This is an automatic collection. Yeah, this is called PAT, and what we are doing is IPCs in process control, which is generally simpler and less costly because that what you see on the right side is very cost intensive. But maybe it has advantages. Yeah, for example, you can use that in production environments to control your reaction automatically. But for that, you need a model, you need calibration. Yeah, and that's the difference to IPC, what we have seen before, because if you need a model and calibration, you're not free as a scientist to develop your reaction in the direction you intend to. So the classic PAT is much more, let me say, tied to a use case. So IPC can cover a wider variety and does not require calibration or modeling. That's the difference. Yeah, okay, let's go to data science. We're going to the end right now. And over to machine learning. Data science means, for example, that you have Python. And this is the example you have seen in one of the slides before, time series. So if you plot the trends we gathered from the database, you have this. And for example, a very modern fashion to, let me say, discuss these curves is the connection to ChatGPT. Yeah, what you can do is, it's very, very easy to understand this program code. You get one of the experiment series. This is the example I always have here. For example, I can have some data, yeah, and I can have the time axis here. Then you go to OpenAI, yeah, connect to OpenAI, and ask on the base of the trends you have imported here, for example, what are the important landmarks in these trends? And with this cliffhanger about the answer, I will finish my lecture here. So I hope it was not too boring. Yeah, it contained ChatGPT, ACD, and ISA-88. Thank you very much, and I'm open for questions. I prefer by chat right now or by LinkedIn. Thank you very much.